number of glia, such as the oligodendrocytes, form a myelin sheath in the central nervous system, which helps speed up the conduction of neural impulses, allows us to think a lot faster on our feet. In addition, we saw how neural lemocytes, which are more commonly known as Schwann cells, form the myelin sheath in the peripheral nervous system. And the myelin sheath in the peripheral nervous system also speeds up the conduction of neural impulses. But there is such a thing as unmyelinated axons in the peripheral nervous system. But even unmyelinated axons have Schwann cells associated with them. But the Schwann cells don't wrap around and around and around like you would if you were winding electrical tape around a wire. Instead, in unmyelinated axons, the axons fit into just small recesses in the surface of Schwann cells. And so they're not tightly wrapped with the Schwann cell to form a myelin sheath. And as a matter of fact, some Schwann cells, and this is an example of a Schwann cell right here. This is what's called a mesaxon. And so right here and right here, right here, right here, right here, right here, those are all axons of unmyelinated neurons. And they fit into small grooves in the surface of the Schwann cell. But like I said, the Schwann cell doesn't wrap around and around very tightly around the axon. And so even though they sit in these grooves on the surface of a Schwann cell, since it doesn't wrap real tightly around the axon, it doesn't do as much to speed up the conduction of neural impulses. So the conduction of neural impulses in unmyelinated axons is on average about 6 or 7 times slower than the conduction speed of myelinated axons. Okay. So this was something that I learned not too long ago. I didn't realize that unmyelinated neurons are also associated with Schwann cells. I just assumed that since myelinated axons have Schwann cells associated with their axons, I just assumed that unmyelinated ones did not. But I was wrong. They actually do. But the Schwann cell doesn't form a myelin sheath. It may be associated with the axons, but it doesn't form a myelin sheath around the axons. Okay. That's just your bit of trivia for today. Okay. All right. So the speed at which a nerve signal travels along a nerve depends on 2 factors. The first is the diameter of the fiber. And the second is the presence or absence of myelin, or a myelin sheath. Okay? So, in general, larger diameter axons conduct signals faster than smaller diameter axons. So the larger the neuron, the faster the conduction of impulses. And also the presence of myelin can further speed up signal conduction. So that has caused some biologists to ask the question, well, if large axons can conduct signals a lot faster than smaller ones, why aren't all neurons huge? Okay, because that would allow us to be able to react more quickly to stimuli. And I don't know that anybody really knows the answer, other than large neurons would take up a lot more space, and it takes a lot more energy to support them and to develop them in the embryo. So maintenance cost of doing so might be a lot higher. in any event, both myelinated and unmyelinated axons actually conduct signals pretty fast. It's just a matter of milliseconds, not even seconds. So the speed at which signals are conducted is basically fast enough. It's fast enough for what we need to do. Note that the length of the axon is not a factor, because conduction speed or velocity is independent of distance, because velocity is equal to the amount of time it takes for a signal to go from one point to another divided by the distance. So conduction speed or velocity is the amount of time it takes to go a certain unit of distance, like per meter. It's like meters per second. Okay, so it's reported as the time it takes over standardized distance. So distance is not a factor in velocity. All right, now in a little bit we're going to take a look at how neurons in the peripheral nervous system can sometimes be repaired by the body or regenerated. And in order to do that, you have to have a myelin sheath, and you also have to have a myelin sheath made up of Schwann cells, and you have to have a neurolemma. There are no neurolemmas. A neurolemma, you have to have an endoneurium. I'm sorry, an endoneurium. Endoneurium is a connective tissue covering that surrounds each of the nerve fibers. There are no endoneuriums in the central nervous system. And so neurons in the central nervous system cannot repair themselves if they're damaged. Once a neuron in the central nervous system is damaged, it's permanently damaged and it becomes permanently non-functional. And another reason why it's thought that brain tissue can't repair itself in the brain once it's been damaged is at one time it was thought that was partly because there are no stem cells in the brain. Since neurons cannot divide, a healthy neuron cannot divide to replace neurons that have become damaged in the brain. That's why once you get a stroke, you usually don't completely recover that because your brain tissue can't heal itself. But more recently, actually, researchers have discovered stem cells in the brain. But they apparently don't have much of an ability to produce new neurons. So it is still true that once a person has brain damage, that part of the brain is probably never going to be repaired. So the brain has a very little ability to regenerate neurons that are damaged. But in the peripheral nervous system, nerve fibers can be repaired or regenerated as long as the soma of the neuron remains intact. The soma is the cell body of the neuron. And at least some of the neurolemma remains. The neurolemma includes the myelin sheath along with the endoneurium, which is a connective tissue covering that surrounds the myelin sheath. So you have to have both of those in order for a neuron to be able to repair itself. If the soma is damaged, or if the axon is damaged so much that there's really not much of the myelin sheath left, then a neuron's not going to be able to repair itself. So this is a diagram right here of a healthy motor neuron. Okay. Now earlier we went over the different structural types of neurons. I think we did that towards the end of last week, actually. So what would be the name for this particular structural type of neuron? There's 4 different structural types. What's the structural type for this one? The functional type is a motor neuron. What's the functional name for that, or the structural name for that? I'll give you some choices. Okay, the 4 different structural types are multipolar neuron, unipolar neuron, bipolar neuron, and anaxonic neurons. Those are the 4 structural types. So which one would this be? It's multipolar, that's right. Multipolar. Because it has numerous cytoplasmic extensions. It has one long one and a bunch of short ones. So what's the name of the shorter ones? The shorter cytoplasmic extensions here. Okay. We went over that too. I think that would have been towards the end of last week. Yeah. Dendrites, yeah, those are the dendrites. Okay, good, you must be studying. Or have a good memory. Okay, so yeah, those are dendrites. Dendrites are the receiving end of the neuron. That's the part of the neuron that's stimulated either by a sensory stimulus, in the case of sensory neurons, or if it's a motor neuron, it's where it gets stimulated by a neurotransmitter that's released by another neuron. Okay. So let's say that this neuron, and these are some muscle fibers over here, and keep in mind that one neuron typically stimulates anywhere from several to possibly over 100 muscle fibers, okay, because they have many branches. All right, so and once one motor neuron is activated, all of those muscle fibers that it supplies are going to contract. It's going to activate all of them. It's sort of all or nothing. All right. So let's say this neuron gets damaged in the middle. Okay, it got cut. Maybe somebody's arm got cut accidentally with a knife or something. That's gonna prevent those neural impulses from being conducted past that damaged point. So that neuron is not going to be able to stimulate those muscle fibers to contract. Okay, now as long as you have part of the myelin sheath and the endoneurium, which is a connective tissue covering that surrounds the myelin sheath, then the neuron may be able to regenerate itself, may be able to repair itself. So the first thing that happens once a neuron is damaged or an axon is damaged is the axon that is beyond the point of damage will degenerate. It can no longer support itself if it's no longer being stimulated by neural impulses. So, but the neural lemma, which is made up of the Schwann cells and the endoneurium, that will remain intact. Those are able to maintain themselves even if the nerve fiber is cut. Okay, so degeneration of the severed axon is the first process in repair. Then the end of the axon that's still attached to the soma begins growing many branches. It doesn't grow just one branch that goes back to replace the missing part of the axon, but it grows a number of branches. Eventually, one of those branches starts growing down the middle of that neural lemma, that sheath that is made up of the Schwann cells and the endoneurium. And this neural lemma then functions as what's called a regeneration tube. So it's sort of a guide that guides that new branch back towards the muscle fiber. Okay. Now, once the once that new axon forms a synaptic contact with the muscle fibers. And a synapse actually does have a small gap between the synaptic knobs and the muscle fibers. So the neuron doesn't actually touch the muscle fibers. There's always a small gap called the synaptic cleft. Once that happens, then these other growth processes that didn't travel through that regeneration tube, those start to atrophy and they start to degenerate. And so that eventually, once those growth fibers degenerate, and then you have a repaired neuron that's able to, again, stimulate those muscle fibers and cause them to contract. Okay. Now, if this repair process does not occur because there's just simply too much damage, there's not enough of regeneration tube left over to allow the repair, then what happens eventually, if muscle fibers are not continually stimulated by neural impulses, they will atrophy. They will start degenerating. And that's referred to as denervation atrophy. Okay. So for a muscle to stay healthy, it has to be continually stimulated. Okay. Now that brings up a question. I just thought of this. I don't know why I did, but it makes me think, if a muscle starts to atrophy, if it's not continually stimulated by neural impulse, what I'm wondering, and I don't know the answer to this question, maybe some of you do, but could you artificially stimulate a muscle with electrical stimulus in order to prevent that from happening? Does anybody know if that's possible? It might be, I don't know. I don't know. It may not work. It may or may not work. I don't know. I don't know if that's possible or not. It's just a question I had in my mind. Might be a good experiment to do on somebody. It may be, though, that you actually need the neurotransmitter stimulating the muscle in order for the muscle to stay healthy. If you just electrically stimulate the muscle, you're not releasing a neurotransmitter on that. So it may or may not work. I'm not sure. So something I may have to look up. All right, let's do a couple more review questions. I asked you a couple of rhetorical questions. Let's do another one. So it says, what type of neuroglia helps form the blood-brain barrier? This has to do with the function of one of the 4 neuroglia that's found in the central nervous system. I count 26 people here, so shows 20 people have answered. Now we have 24, so we still have 2 more people. So go ahead and take a guess if you haven't already answered. All right, so let's take a look at all your answers. A little over half said astrocyte, and that's correct. Astrocytes are neuroglial cells in the central nervous system. They send out cytoplasmic extensions. Some of them make contact with blood vessels in the brain, and they induce the blood vessels to become less permeable, particularly to drugs and toxins. It's not a perfect barrier, though. there are some substances that are allowed to pass through the blood vessels. Obviously, you would have to allow oxygen and nutrients to diffuse out of the blood vessel. But it does prevent some drugs, especially therapeutic ones, to get into the brain tissue. So that makes it difficult to treat certain diseases. Okay, here's another one. Okay, what's the name of the neuroglia that helps to fight infection in the brain? Actually, I'm going to mention something after you get done answering here. This particular cell that this question is referring to, they think it might be associated actually with Alzheimer's disease. Not the presence of it, but a lack of sufficient numbers of this cell type. Okay, let's take a look at the results. Most of you said microglia, and that's correct. So this is the cell. There's some researchers that have noticed that patients that have Alzheimer's disease, which causes dementia, some of them have lower than normal numbers of microglia. And one of the signs of Alzheimer's disease is plaques develop in the brain, and that's thought to prevent normal nerve conduction. They think that some of these microglia may help remove those plaques to prevent Alzheimer's disease from occurring. People that have Alzheimer's disease may have too few microglia, or maybe microglia that don't function as well as they should, and they're not able to keep those plaques removed. And they think that may be one of the reasons for it. That's just a hypothesis at this point, though. It hasn't been proved. All right, so that's the end of this section of chapter 12. So I want to go ahead and move on to the next section, which is titled Electrophysiology of Neurons. And whoops, did I open up the wrong one? Did I open up the wrong one? That's the same. Shoot. Oh, here it is. Here it is. Okay. Okay. So, two of the terms that I want to introduce today that have to do with conduction of neural impulses. Yes?
After class, could you upload this?
Oh, yeah, I'll do that. Yeah, I have it on there. I'm sorry, I forgot to publish it before class. So I was working on getting a sign-up sheet for advising on my door. Okay, so electrical potentials and currents are two terms that relate to the production of neural impulses. And so the transmission of nerve impulses is due to the presence of electrical potentials across the cell membrane in axons of neurons. And electrical potential is a separation of charges across the cell membrane. So the charges that are outside of the membrane tend to be more positively charged. And on the inside of the membrane of the axon, there's an excess of negative charges. And so that difference in the two types of charges is called electrical potential. And this charge difference across the membrane of the axons is due to only two factors or variables. The first one that makes this possible are the concentration gradients of sodium and potassium on the two opposite sides of the membrane. This concentration gradient of sodium and potassium is due to the activity of the sodium-potassium ATPase pumps, which is a form of primary active transport. There are sodium-potassium pumps in the cell membrane of all cells, including neurons. And they're constantly pumping sodium out and potassium in. Okay. And it's because of those sodium-potassium pumps that there's a concentration gradient of sodium and potassium across the membrane, where sodium is higher on the outside of the cell, on the outside of the membrane, and potassium is higher on the inside. However, potassium has a tendency to leak back out of the cells because there are what they call potassium leak channels in the cell membrane. So potassium just leaks back out, and when that happens, that leaves an excess of negative charges on the inside of the cell. So this is one factor that makes this electrical gradient or electrical potential possible. The other factor is the relative permeability of the membrane. As I said, the membrane is more permeable to potassium because of the presence of potassium leak channels that allow potassium to leak back out again. And so that's what makes it possible for the inside of the membrane to be more negative than the outside. Sodium, on the other hand, is not very permeable in the membrane at all. Sodium ions are positively charged. And remember, the middle of the membrane is a nonpolar environment. The middle of the membrane is made up of fatty acid chains. And those fatty acids are nonpolar. Polar substances don't mix very well with nonpolar substances. So sodium can't go through the membrane very easily at all because there aren't any sodium leak channels in the membrane. The membrane of neurons does have potassium leak channels. So that makes potassium about 1,000 times more permeable in the cell membrane of neurons compared to sodium. So anytime charges are separated across a semi-permeable barrier like a membrane, then that membrane is said to be polarized. Okay. Now, if we allow some of those charges, like the sodium ions, which are positively charged, to move across the membrane by somehow changing the membrane's permeability, then that causes an electrical current to flow. Okay, so electrical current is the flow of charges from one point to another. So if we allow sodium to move across the membrane, that's producing a current. It's producing a current of positive charges across the membrane. And when that happens, the membrane is said to be depolarized because those positive charges, when they move across the membrane, they'll neutralize the negative charges on the inside of the membrane. And so it's no longer polarized, it's been depolarized. Okay? So let me show you an example of this. Let me show you a little model. Okay? So this is just a very simple model of the membrane on one side of the axon. So this solid line right here, that represents the cell membrane. Okay, so this is just a very simple schematic diagram. These positive charges are on the outside of the membrane. And those positive charges are caused by the excess of sodium on the outside of the membrane, because the sodium-potassium pumps are pumping the sodium out of the cell. These negative charges on the inside, that's on the inside of the axon, that's due to the potassium, which is also positively charged, leaking out of the cell. Since potassium is positively charged, if you remove positive charges from the inside of the axon, that leaves an excess of negative charges. Take away positive charges, it makes the inside more negative. Okay, and so this membrane is said to be polarized because you have a separation of charges across the membrane. So if we do something to increase the permeability of sodium in the membrane, then the sodium ions will be able to diffuse across the membrane for two reasons. They move across the membrane because they're moving down their concentration gradient. So they move across the membrane just due to simple diffusion. And secondly, they're also attracted to the negative charges because opposite charges attract. So if you have positive charges on one side of the membrane, negative, the positive charges are going to want to go across the membrane. They're attracted to those negative charges. Okay, but without doing anything to the membrane to change the permeability, those sodium ions can't do that. They can't move across the membrane, so it'll stay polarized. But if we can stimulate the membrane in a way which changes its permeability, then those positive ions can move across the membrane and it'll neutralize those negative charges. And so the membrane is said to be depolarized. Okay. So that's actually how neural impulses are initiated. They're initiated when the membrane depolarizes. And that causes a change in the electrical potential of that membrane. So that's just the way it initiated. We'll talk later on about how that depolarization moves down the axon. Okay, there's a wave of depolarization that moves down the axon. All right, so it's important to understand the differences in the relative concentrations of sodium and potassium on the opposite sides of the membrane. So, just to check your understanding, which of those two ions is in higher concentration on the outside of cells? So one way you can remember this is just think about how the sodium-potassium ATPase pumps work, because that's what produces these concentration gradients. That and the relative permeability of the membrane to sodium and potassium. Go ahead and put in your answers. So, most of you said potassium is higher on the inside of cells, and that's correct. That's correct. And that's because the sodium-potassium ATPase pumps 3 sodium out of the cell and 2 potassium in. Like I said, that potassium doesn't stay on the inside of the cells because of the presence of potassium leak channels in the membrane. It allows potassium to move down its concentration gradient and diffuse out of the cell. And that's what leaves the inside of the cell more negative. Okay, so one of the situations which makes a neural impulse possible is what's called a resting membrane potential. Resting membrane potential is a separation of charges across the membrane while a neuron is at rest when it's not being stimulated. So this is kind of a schematic diagram that shows the factors which cause the establishment of the resting membrane potential, which happens to have a value of about minus 70 millivolts. Now, the way that membrane potentials are measured is they're measured by, actually, you can stick a very fine probe through the cell membrane of the neuron. And so membrane potentials are always measured on the inside of neurons. Okay, so that's the voltage on the inside of neurons. Since the inside of neurons have an overall negative charge at rest, the resting membrane potential has a value in most neurons that averages about minus 70 millivolts. And I say about because it varies slightly from one neuron to another. That's the average value. Some of them might be a little higher, some of them might be a little lower. Okay. So this resting membrane potential is due to the concentration gradient of ions on both sides of the membrane, plus the relative permeability of the membrane to those ions. So this represents a sodium-potassium ATPase pumps. So keep in mind it does require ATP because it is an active transport process. So that pumps 3 sodium out of the cell. At the same time, it pumps 2 potassium inside. So that's going to cause an excess of positive charges on the outside of the cell because this keeps pumping sodium. Sodium is relatively impermeable in the membrane. It can't diffuse across the membrane without some specific transport mechanism in place. So that causes an excess of positive charges on the outside. But that's partially offset by the positively charged potassium that's pumped into the cell. However, there are leak channels in the cell membrane that are specific for potassium, will only allow potassium to go across the membrane. And so, since this ion pump is continually pumping potassium into the cell, potassium builds up in the inside of the cell. And so you have a higher concentration of potassium on the inside than you have on the outside, so you have a diffusion gradient. And since there's leak channels, this potassium starts leaking back out of the cell. And as that potassium moves out of the cell, if you have fewer positive charges inside of the cell, that's going to make the inside of the cell more negative. So it's the diffusion of potassium out of the cell that causes the inside of the axon to be negatively charged. The positive charges on the outside are due to the excess of sodium that was put there by the sodium-potassium ATPase pumps. So that polarizes the cell, and it polarizes the cell to a magnitude of minus 70 millivolts. Okay. So that resting membrane potential is due in part to the relative concentrations of sodium and potassium across the cell membrane. But there are some other ions that also have an effect on the resting membrane potential, because there are concentration gradients of other ions across the membrane. For example, there are more proteins on the inside of the cell than there are on the outside. Proteins tend to have more negative charges than they do positive charges. So overall, most proteins tend to be somewhat negatively charged. So that makes the inside of the cell membrane even more negative than it would be if it were only caused by the diffusion of potassium. And so the presence of protein inside the cell is going to make that resting membrane potential even more negative than it would be if we only factored in potassium alone. Another ion that has an effect is chloride. Chloride ions are negatively charged, and chloride ions are in a higher concentration on the outside of the membrane. And so that makes the outside of the membrane less positive than it would be if there wasn't any chloride. And so that brings the resting membrane potential down a little bit. So when you factor in all of the different ions that have concentration gradients in the cell membrane, neurons have a resting membrane potential of about minus 70 millivolts. And that membrane potential is what makes neural impulses possible. You have to have a cell that's at its resting membrane potential in order for that neuron to produce a neural impulse. Okay. So what we're going to do next in this section of chapter 12 is we're going to look at the contribution of each one of those ions that I mentioned, like chloride, as well as sodium and potassium and a few other ions, because each one of them contributes in at least a small way to this resting membrane potential of minus 70 millivolts. Okay, so we can do that by looking at the effect of each ion individually, and so the overall membrane potential is actually the sum of the effects of all the individual ions on the membrane potential. But to do that probably is going to take us about 15 or 20 minutes. So we don't have time to get into that today. So this probably would be a good place for us to stop, okay? Because I don't want to get started and have to stop halfway through when we have a whole weekend before we can bring it back up again. So but we'll have to stop here, and we'll start off. What we're going to do is we're going to talk about equilibrium potentials next week.
They thought they had